Hello. Okay, so our next topic, we will be discussing more about the structure of DNA. We kind of started talking about that when we were talking about chemistry and macromolecules, but we didn't finish the story because now is when it's relevant. And so it was kind of like, what's the point of learning all that back then when we're not going to use it until now? So now is the time. Okay. Um, Secondly, we're going to talk a bit about you've heard me say repeatedly that DNA is like the instructions for the cell, right? And we've now seen that when we talk about genetics, right? So we're talking about genes, right? And you inherit different alleles from your parents, and those alleles determine things about you, okay? So we together have discussed that happens, but we have not yet talked about how it happens. And that's where we're going next. Okay? So I like to pepper in a little bit of history. I have to admit, one area where I'm a little bit behind is one area of growth I see in how I teach biology is to talk more about underrepresented groups in science. So unfortunately, you'll notice that a wide range of scientists that we do talk about, and we don't talk about that many in here, right? I don't mention individual scientists' names a ton, but many of them are male, white, usually of European descent, right? Um, and so it's nice to know that there are scientists of other varieties, right? So we're going to do a little of that today. I think that historically, both women and persons of color are historically not as active in many areas of science, but even worse than that, their contributions are not acknowledged as much, right? So even when they have had very important roles in science, instead of getting the acknowledgement that they deserve, often they're kind of ignored. Okay. So anyway, so that's an area where I'm going to be making progress in the future, but we will talk about a woman today. Okay. So determining the structure of DNA, there's a great little video in your homework that talks a little bit about the history of this. One of the reasons why I think this is kind of entertaining is it's fairly recent in the grand scheme of things. I know you're like 1950s is recent. What? But it's more recent than, say, when Mendel or Darwin were doing their research, right? So relatively speaking, this is more recent. In fact, many of you probably know somebody that was alive during this time, right? My parents were born in 48 and 50, right? So I often kind of marvel at the fact that when my parents were born, people had not yet figured out the chemical structure of DNA, right? And it's like, and now here we are figuring out how to like utilize DNA to cure disease, yeah, in my parents' lifetime. And I think that's kind of cool. Okay, so anyway, all right, enough blabbering. So the two people who get the majority of the credit for discovering the structure of DNA are James Watson and Francis Crick. They're a team. You can see them there with their model of DNA. A team of biochemists. Crick is American, Watson is British, and they they got the Nobel Prize for this, and they published in the mid 50s, right, this structure of DNA. There was a big it was almost like a competition to figure this out. There were a whole bunch of different scientists that were working on this. Some people thought, oh, the molecule that's responsible for inheritance, right, for how we get the traits that we get, must be a protein because proteins are super complicated and large. And so it's got to be a protein. That makes the most sense. And other people are like, no, I don't think it's it. And then they're like, okay, so they finally figured out, okay, well, it's DNA, but they didn't know how DNA was structured. Right. So they knew the components as far as like the actual elements that made it up, but they didn't know the shape yet. Which means that they didn't really understand how it worked either, right? They didn't know anything about how it worked. They knew that it was inherited, but they didn't understand how. Okay. And so the interesting story, you'll watch a video about this, is a story about Rosalind Franklin. And so Rosalind Franklin was an X-ray crystallographer. And what that means is that she used X-rays to produce images of molecules, essentially, right? And so she collected data. She was essentially making X-ray images of DNA, right? And Watson and Crick saw her images and basically were like, meh, 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 you know, whatever, not so useful, blah, blah, blah. But it turns out that those images, the data that she collected, was vitally important ultimately for them to figure it out, right? And so these two gentlemen are kind of credited with being the first to discover the structure of DNA, even though lots of other scientists were trying at the time. And Rosalind Franklin very rarely gets any attention, which is, you know, it's unfortunate because she played an incredibly important role in all of this. The first time I talked about this in class, one of my students was really irritated that when you see images of these scientists, right? When we see Francis and Crick, they're looking all sciencey with their science model and, you know, being all scientists. Yet Rosalind is, you know, posed like this with her pearls and, you know, looking like she's, you know, it's a yearbook photo or something. And it's like, why doesn't she look like she's doing science? Why is she only being valued for her appearance in this picture? And I was like, dang, Skippy girl. And then so she sent me some images, right? So here's Rosalind actually doing science, right? So there she's using a really cool old microscope, right? There she is weighing things out, right? Wearing a dress, because that's what women did in the 50s, even if you were doing science, right? And here is one of the images that she produced, okay? And so if you're looking at this and then you're up here looking at this, you're like, I don't get it, right? This essentially is an image of if you were looking down into a double helix of DNA. Okay. So anyway, watch the video in the homework. It's really interesting, right? And it kind of gets to the idea. I also really like it because it gets to this idea of this like humanizing aspect of science, right? The people who are studying these things and learning these things are humans, right? And they have human lives that involve more than just science. So it's kind of interesting to learn a little bit about the history and relationships of these people. Okay, so that's the team. These are the people that figured it out. It turns out Watson is a not a good person. He's a raging racist, as it turns out. I don't know. I haven't heard much about Crick. Rosalind Franklin initially did not receive any, you know, sort of acknowledgement of her contribution until after she died, because she was exposed to X-rays, right? And at the time, we didn't fully appreciate how dangerous radiation was for people's health, and so she died of cancer, like a lot of people, a lot of scientists who were doing science using X-rays. And yeah, so she died young, right? She died in her 40s, I believe, might have been her early 50s, as a result of cancer caused by working with X-rays. So the very tool that she was using to help make these contributions is ultimately what killed her. And because the Nobel Prize is not given posthumously, right? She never got the acknowledgement she deserved. So angry. All right. Anyway. So watch that video, it's kind of interesting. All right, so now let's talk about what they all actually discovered, okay? So DNA, maybe we should write that out, deoxyribonucleic acid. Okay, DNA. Deoxyribonucleic acid is one of the nucleic acids. So of the 4 macromolecules we talked about, right? We talked about carbohydrates, lipids, proteins, and nucleic acids. Okay, so this is the 4th one. And there's 2 different types of nucleic, well, there's more than 2, but there's 2 main types of nucleic acids that we talk about. One of them is DNA, and the other one is RNA. And so we'll talk about RNA in a few minutes, not yet. Okay, so just like our other macromolecules, DNA is made of individual units that we call monomers. Remember that? Right, so single unit, and then it combines many monomers. Excuse me, I'm like gonna sneeze or yawn, or you'd think I would know how to use the like pause button. Okay, whew. Made of many units, okay, those individual monomers collectively called a polymer. Okay, so the polymer technically is called a polynucleotide, and it's made up of individual units called a nucleotide. Now, the way that it's arranged, DNA is a double helix, right? So helix just means that it's a twist, okay, which you can see here. It's like a twisted ladder. And double because it has it has a strand on either side that's then twisted, okay? This little outline here, this dashed line, surrounds a single nucleotide. Okay, so I'm going to erase where I wrote just because it's kind of cluttering up the picture, right? But see this little bracket right here? So this is one nucleotide. So each nucleotide has 3 parts. So here's a single nucleotide. We're like zooming in. We'll come back to that previous picture in a second. And so the 3 parts of a nucleotide are a phosphate, a sugar called deoxyribose, which is not surprising. Okay. Yeah, so we got this sugar. We have a phosphate group, we have a sugar, and then we have a base. Sometimes these are also called nitrogenous bases. And they're called that because they have nitrogen in them. Go figure, okay? You do not need to know any of this detailed structural chemistry, right? So like you don't need to draw what a phosphate group looks like. You don't need to know what the deoxyribose looks like. You don't need to know the structures of the different bases, but you do need to know the names of the parts, okay? So, right, the 3 parts are phosphate, the sugar, the nitrogenous base, and then there are 4 of those. Right? So here they're just kind of showing you like a, gray hexagon to stand in the place of those 4 possible bases. Okay, and so the bases are called adenine, guanine, thymine, cytosine. This is the A's and T's and C's and G's that you may be familiar with. You may have heard of these before. Okay, you'll notice that A and G are both double ring structures, so they're kind of fat relative to the size of thymine and cytosine, which are single ring structures, so they're skinnier. Okay. All right, let's go back up to this for a second, okay? So what we're looking at in this picture now over here is a simplified version of this, right? And so the yellow, blue, and then the different colored bases, yellow, blue, different colored bases, okay? So the sugar and the phosphate make up the rungs of the ladder, if you want to think about it that way. I shouldn't say the rungs, I should say the rails of the ladder, right? And then the individual rungs of the ladder, the parts you climb up when you're climbing a ladder, are made up of the bases, okay? Now, bases pair in a particular pattern. So I typically refer to this when I'm talking about this for the lab you're going to do for this material. I talk about the base pairing rules. Essentially what that means for DNA is that A and T, adenine and thymine, always go together. And C and G, guanine, cytosine, always go together. Okay, so the nice thing is that they've color coded these to reflect that. So the green ones go together and then the, orangey ones go together. But let's talk about why they fit together, okay? So first of all, notice how they're held together. This is kind of interesting. So do you see those little dashed lines? Here, 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 right? Those are hydrogen bonds. So if you recall all the way back to learning about chemistry, right? And learning about the different types of chemical bonds, right? Remember that hydrogen bonds are fairly weak, right? This is why I can slosh my water around in my water bottle, right? Because the bonds will break and then reform. So it's fairly weak, but it's significant, right? So it holds these things together, but it doesn't hold them together super duper tightly, which is useful because your cell needs to be able to separate those two strands of DNA. Okay, so one thing that I hope you notice by looking at this diagram is that the A's and T's are held together by two bonds, two hydrogen bonds, 1, 2. Whereas the C's and G's are held together by 1, 2, 3. So it's highly unlikely that, I shouldn't say highly unlikely, I should just say regular unlikely. It is unlikely that these will be matched up incorrectly because they fit together like a puzzle. Okay, so the reason, I'm losing my mouse. Oh, my computer's not happy right now. Come on, mouse. Okay, can't use mouse, must use touchpad. So the reason that A and T go together is because they fit together like puzzle pieces. They each are able to form those two hydrogen bonds. C and G are able to form those 3 hydrogen bonds, okay? Another thing you'll notice about each of these pairs is that each pair is composed of one of the double rings and one of the single rings, right? So, yeah. Come on, mouse. What's happening to you? Is your battery dying? Am I overusing you? Yes. So A is a double ring structure. T is a single ring structure. They go together, right? G is double ring, C is a single ring. They go together, okay? What do I want to say? Yeah, right? And so what that does is that maintains the width of the double helix so that it's not like super fat in some areas and super skinny in other areas, okay? So it very much is structure, right? They fit together like puzzle pieces, okay? And so when you look at images like this over here, right, they often show the A and the G as being longer, like over here, right? The A and the G are longer than the C and the T because they're double rings instead of single rings, right? And then they show these little notches to show how they fit together, and isn't that cute? Whatever. Okay, so that's the structure of DNA. As you recall, there is a lot of DNA in your cells, okay? So depending on what kind of organism you are, right? For humans, our DNA is found in strands that we call chromosomes, and we have 46. Yeah, this reminds you of that. If you were a prokaryote, if you were bacteria, it'd be a single circle, right? So that's chromosome. Chromosomes are essentially strands of DNA, all right? And all the term that we use to describe all of the DNA together, the full set of DNA, is the term genome. I don't know how much we've talked about this up until this point. I don't recall if I've mentioned that word at all, actually. Okay, so genome refers to, you know, basically the entire set of all of your DNA is your genome. Okay, the term gene, we have talked about this, describes a section of DNA that codes for something. So typically, right, a typical gene is about 3,000 nucleotides long. Okay, but it varies. Some are shorter, some are longer, right? But it's a chunk. Okay, and what we'll be talking about in the next couple of weeks is how that gene codes for a protein, okay? Now, we'll talk about the fact that, as it turns out, not all genes code for proteins. It's not as simple as textbooks make it sound, which is frustratingly misleading. Okay. But that's sort of the model that we're going to use to start. So for example, like remember when we were talking about cellular respiration and we said, okay, this is how the cell breaks down glucose. And then afterwards it was like, yeah, but remember that, you know, you use more different molecules for energy other than just glucose, right? So it's going to be one of those conversations where it's like, okay, we're going to talk about how DNA codes for a protein. And then later we're going to talk about sort of the exceptions to that and, you know, how it's really more complicated. Okay. All right. So where do I want to stop? All right. So here's what we'll do. Okay. Let's talk about DNA replication to start with. Okay. And then we'll stop after this. So one of the really tricky things about understanding the structure of DNA, and I didn't even talk about the people that were involved in discovering that DNA was the compound that was responsible for inheritance. Okay, I didn't even talk about that study. That's a whole different thing. Okay, different people, different question. Okay, but they knew that the DNA had to be replicated somehow, right? How do you make a new cell with a full new set of instructions? Well, you have to be able to replicate it. And this was the part that was like confusing everybody when they were trying to figure out the structure of DNA. They were like, we don't, what, huh? How does that, what? Okay, because it's kind of tricky when you're trying to discover something that we don't already know about. Okay, for us it's not as complicated because we know. So I can just tell you. Okay, all right, so this is meant to be sort of a schematic diagram that shows what's happening with DNA replication, and then we're going to look at the nitty-gritty details in a little bit more detail. But before we even get into that, we need to like hold up for a second, and we need to talk about when replication happens. And right now you should be yelling at the screen. You should be like, I know. We learned about that in chapter 5. When does replication happen? When does DNA replication happen? That's right. It happens in interphase. Okay. So we know what happens in interphase because we talked about that before. Now we're going to talk about how it happens. Okay. So keeping in mind that DNA is this helix, right? That's shaped like a ladder that's been twisted, like a rope ladder that's been twisted. Okay. The two sides are complementary to each other, right? So I mentioned the base pairing rules earlier. Notice this is our original DNA molecule here. Notice there's two strands. And notice that our base pairing rules apply. So if it's T on this side it's going to be A. If it's G, it's C. If it's A, it's T. So on and so forth. Okay? This is how information is stored in DNA. Only those 4 letters. It's kind of amazing. So what order they're in, what the arrangement is what matters for how genes are used. Okay? So in DNA replication, essentially what happens to, you know, sort of oversimplify the whole thing is you have this double helix, right? And so what you do is you grab the 2 strands and you pull them apart. Okay? Because remember, they're only held together by hydrogen bonds, and it's relatively easy to break those apart. Okay? So we pull them apart, and then free nucleotides that are just kind of floating around come in and use each side as a template to make 2 new double helices, is plural for helix. Okay, so if we take this blue double-stranded dude and we separate it, so this strand goes over here, this strand goes over here, okay, then newly synthesized DNA, so DNA strands from the original molecule are in dark blue, and then the new ones are this kind of teal color, turquoise, I don't know, whatever it is. Okay, so this acts like a template so that individual nucleotides can build on that match. Okay, so if we got an A here, we get a T. If we have a C, we get a G. If we have a T, we get an A. You see where I'm going with this? Okay. Now this side also acts as a template. So if it's a T, you get an A. If it's a G, you get a C. If it's an A, you get a T. Okay, so what do you notice now if you compare this new but really it's only half new, right? Because one piece we had before, okay? So our new DNA molecule over here and our other new DNA molecule over there, how do they compare to our original? Oh, look at that. They're exactly the same. What? Okay. So the difference here is just that this is like flip-flopped, okay? So our original one is exactly the same as the bottom two because it doesn't matter which orientation is which. So you might be looking at this and saying, well, the left strand on this one starts with T and it's the right strand that starts with T on these. It doesn't matter because remember, this is all just floating around in the cell. So it doesn't matter which way it's facing. I feel like Beyonce. Okay. Yeah. Does that make sense? Okay. So these two new strands, right, are identical to the original DNA. Okay, so that in a nutshell is what's happening in DNA replication. This is more like what it looks like though, right? Because remember it's a double helix, right? So that double helix has to be pulled apart, right? And then each side acts as a template for more DNA to get built, and you end up with two exact copies, right, of the DNA that are exactly the same. Okay. This happens incredibly fast, right? Incredibly fast, right? So entire chromosomes are copied in like, seconds. Okay. It happens very, very quickly. Partly because it doesn't just like start on one end and then go all the way to the other end. That would take forever, right? Because they're long. Remember, these are really, really long. So the way that it happens is it happens in these little spots they're called the origin of replication. They kind of look like little bubbles where the DNA separates in multiple different places, right? And then grows from both directions. So it doesn't just start on one end and go to the end. It would be like if you were trying to pull apart, I don't know, string cheese, but instead of your string cheese being this long, your string cheese was like, 4 feet long, right? If you peeled one end and like peeled all the way to the other end, it would take forever. But if a whole bunch of people all grabbed one little area of the string cheese and pulled it like that, you could pull the whole thing apart in, a fraction of a second. Yeah. So that's what these do. Okay. So it separates the two original strands and then builds the new. So in this image, this is not in your book, the blue strands are the original DNA and then the orange are the new strands. I don't like that it says parental and daughter. I think that's really confusing when they say that because anyway, but old and new. Let's say that. Okay, so the orange is the new. Okay, little more detail. So here's a picture of what that looks like, right? So we call it a replication bubble. So our original double-stranded DNA is pulled apart, right? You have this little bubble, and whoa, that picture's ugly. Ah! Okay, so take a deep breath. Don't look at the picture for a second. I'm just going to tell you a story, okay? In order for anything to happen in your cells, almost anything to happen in your cells, enzymes need to be involved. Okay, so it's been a little while since we talked about enzymes, so maybe let's have a quick refresher about that. So enzymes are a type of protein, and enzymes are responsible for speeding up chemical reactions. Right, and so remember that in the process they are not used up. Right, they assist in sort of allowing a chemical reaction to take place, but they don't get used up in the process. Okay, so we have this enzyme, and this enzyme is called DNA polymerase. Okay, part of the reason that I know it's an enzyme is because the letters ASE are used all the time in enzymes. So enzyme names often end with ASE. So from now on in your life, whenever you hear blah 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 ACE, right, you should go to yourself. Just wait, right? If it's biochemistry that we're talking about, wait, blah 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 ACE. That's probably an enzyme. Okay, so DNA polymerase is an enzyme. Based on its name, what do you think it does? So the other part of the word we got here is polymer. Polymer. You know what polymers are, right? They're linked together monomers. So guess what DNA polymerase does? It is an enzyme that builds polymers of DNA. Okay. And so there's the reason this picture looks all weird and blobby is they're trying to show you like the actual structure of this. Okay. They're trying to show you like, you know, more or less what DNA polymerase actually looks like. Okay. So, right. So this big orange and brown blob is the DNA polymerase. Now, going back to the colors we were using before, right, the dark blue and the turquoise, okay, so the dark blue represents the original strand, double helix of DNA, okay? And what the DNA polymerase does is it hops on there and it kind of pulls the strands apart so that, and then it brings in the appropriate matching nucleotides, right, to match up with, right, the original strand. And ultimately what this is producing this, right? Okay, that's what it actually looks like. There's videos of this, okay? Now, remember that this is happening, hold on. I got a book here. This is my little fun. I use this all the time in my other classes. I haven't made you guys suffer through this at all, really. So one of my favorite old books that I have is a dictionary of root words and combining forms, which has all the Latin and Greek roots for everything, which I tell you guys a lot, right? I tell you this is what this word means, this is what this word means, right? But sometimes I like actually looking them up. So ligase, let's talk about that. So what can you tell me about DNA ligase? Well, the ACE tells you it is an enzyme that probably does something with DNA because that's in the name. So let's look up LIGA, shall we? Let's look up LIGA in my book. LIGA is Latin for bound, tied, a band. Okay, so what DNA ligase does is it basically sticks together those pieces of DNA. Okay, it's cool, right? Yeah. All right, so that's another enzyme that's useful. Okay, so it sticks together those pieces. Okay, so that, where do I want to stop there? Yeah, I think that's where I want to stop. Okay, so that's DNA replication in a nutshell, in a very simple way. Okay, so two important enzymes that are used, right? DNA polymerase pulls them apart so that the complementary nucleotides can jump on to the two original strands. And then DNA ligase comes along and it basically just kind of like sticks, I'm oversimplifying because that's what I do, kind of sticks everything together. Okay. All right. Okay. So that's DNA. That's your intro to DNA and DNA replication. Okay. And then we're going to have two more videos. One of them is going to be about transcription. The other one is going to be about translation. Okay. Do I want to talk about RNA now? Yeah, I might as well. Okay. No, I'm going to save that for later. I'm going to save that for the next video. Okay. All right. Done.